Welcome to the medical classroom with Gerald, and today we shall be talking about gametogenesis. But first of all, you will need these 4 textbooks as you're seeing them on the screen. Meanwhile, click that subscribe button, like the video and comment, and support us to enable us to reach further. We shall begin with the definition, and then we shall talk about the chromosomes, we shall talk about meiosis, we shall also talk about oogenesis, and then spermatogenesis. This gametogenesis is always the first topic you cover in histology and embryology as far as year 1 histology and embryology are concerned. So you need to understand it fully and be able to answer respective exams when they are set from this section. As we see, gametogenesis comes from 2 words, gametes and genesis. Genesis meaning beginning, so we are forming and developing gametes. And remember, we are having male and female gametes. So this is formation and development of gametes. As simple as that. A process that leads to formation of the female gametes is the oogenesis, and the ones forming the male gametes is spermatogenesis. And along the way, we need to know about chromosomes and this division, meiosis, how they aid us in formation of these gametes. You also need to note that the human body has 23 pairs of chromosomes, and 22 here are autosomes. They are somatic cells. And then 1 pair are the sex chromosomes. So after knowing that, we need to know the structure of a chromosome. So the chromosome structure varies depending on DNA replication. Before replication, a chromosome exists as a single chromatid chromosome. This is before replication, before DNA replication. So we have this center part, what we call centromere. We have these 2 chromatids. Now one is always a short arm, or what we call the P arm. The one on top, and then we have the long arm, or the Q arm down. And these 2 are interconnected by the centromere. So after DNA replication, chromosomes are double, are chromatid chromosomes. That is briefly about the chromosome. So these chromosomes have types according to the location of this centromere that you also need to really check out. The first one is metacentric. We have submetacentric. We have acrocentric, anterocentric. You need to know these 4 types of chromosomes. Then let's remind ourselves of what we meant by homologous chromosomes. Remember, these are chromosomes that are of the same size. They are of the same shape. They are of the same length. And that share the same gene location known as loci. So take an example of these 2 chromosomes. Remember, we are saying they are of same length and same gene location. So after DNA replication, we form what we call sister chromatids. So these are sister chromatids, these 2. And these 2 are also sister chromatids. Then we also need to know that normal somatic cells and primordial germ cells contain 46 single chromosomes, and these 46 single chromosomes occur in pairs of 2, so those are the 23. And on each pair, we have equal contribution from maternal origin and from the paternal, from the father. And we also need to know that these somatic cells have 2n amount of DNA. So let's briefly answer these terms. What is a diploid cell? It is that cell that contains 46 single chromosomes. And the haploid contains now half the number. So it contains 23 single chromosomes. And what is ploid? This is the number of chromosomes in a cell. And what is the n number? It is the amount of DNA. In a cell. That is briefly what we need to know about chromosomes. as gametogenesis is concerned. Now let's go to meiosis. So we all know we have two types of meiosis. We have meiosis 1 and meiosis 2. And in this cell division, we have main three results. One, we have formation of haploid of gametes, 4 gametes. And these gametes contain half the normal number of chromosomes, meaning they contain 23 single chromosomes. And these gametes have half the amount of DNA. So they will have 1N. So we are saying this meiosis results into formation of cells of this kind. 23 single chromosomes with half the amount of DNA. And then you'll be asked about the main events that occur in meiosis, and these can't miss. Let us first outline them. The first of all, we have synapsis. And from synapsis, we have crossing over. From crossing over, we have alignment. And from alignment, we have disjunction. And from disjunction, we have what we call a final step of cell division. So we have synapsis, crossing over, alignment, disjunction, and cell division. These 5 events have to be at the back of your mind. And what happens in synapsis, we shall have pairing of those 46 homologous duplicated chromosomes. And in crossing over, here we shall have exchange of large segments of DNA. You remember those questions in our A-level about crossing over, which is one of those factors leading to genetic variations in populations. In alignment, we shall have these 46 homologous duplicated chromosomes aligning at the base at the metaphase plate. And then in disjunction, we shall have their separation. And you know that the moment we have non-disjunction, we are bound to have chromosomal abnormalities. And we shall also see it in the anomalies that come in this gametogenesis, disjunction, where we shall see we are having children with microcephaly, those disorders. And here in cell division, we shall form two secondary gametocytes. And these gametocytes will have 23 duplicated chromosomes, and these chromosomes will be of 2n. You have to know that in meiosis 1, we are forming 23 here, but with the 2n. The amount is not yet halved. We are having synapses. They are pairing. Then we are having crossover, they are exchanging the genetic material. We have alignment, and then we have disjunction, and then we have cell division. So these steps follow each other, and in case they come in objective questions, you should be able to answer them. So let's go to events that occur in meiosis 2. So let's begin with synapses. In meiosis 2, we have no synapsis, is absent. Should note these for your objective questions. How about crossing off? In meiosis 2, we have no crossing off, but we have alignment. Yes, we shall have alignment of these 23 duplicated chromosomes. At metaphase plate. How about the disjunction? Yes. We shall have separation of these 23 duplicated chromosomes. And they will be forming 23 single chromosomes. So then cell division, it's going to give us 2, then plus the other 2, those are 4 gametes, and these gametes are going to have 23 single chromosomes, 23 single chromosomes, and these chromosomes are going to have half the amount of DNA, such that when you take this and you meet a man or a woman, after fertilization you have 46 and 2n, the normal diploid number. So that is briefly what you need to know about meiosis, and we shall see more of it in the subsequent processes. So for exams, you've already known the definition, you've seen the chromosome before DNA replication, after DNA replication, we've seen the types, and we've seen the main events. We've also realized that we don't have synapsis and crossing over in meiosis 2. And after meiosis 1, we are forming 23 duplicated chromosomes, but these chromosomes still have the original amount of DNA. But after meiosis 2, they have half the amount, and these are 23 single chromosomes. Let's talk about oogenesis. Oogenesis, briefly, it is the formation of female gametes and it occurs in the ovary. This is one of those simple processes and questions that come here. If you've read, they tend to be very, very simple. So we need first appreciate that the primordial germ cells that lead to formation of these female gametes, they come from epiblast. So briefly, around your week 2 of development, we shall have epiblast cells. Some of these cells are going to differentiate into your primordial germ cells. I tend to call them PGCs. So these primordial germ cells are going to migrate to the wall of yolk sac, and this is always around in the 3rd week. And then in the 4th week of fetal life, they arrive at the ovary. At the 1st week, from your epiblast to the ovary. So when they arrive here, they are going to differentiate, and they differentiate into cell types called oogenesis. So your oogenesis are going to populate the ovary. And you also need to note the chromosome number and the amount of DNA. And these primordial germ cells are 46, 2n, and they are also forming oogenesis, which are 46, 2n. So these oogenesis, they are going to divide and they will undergo DNA replication to form primary oocytes. And all these primary oocytes are formed by fifth month of fetal life. I think I forgot to air out that this process, oogenesis, starts before birth. As compared to spermatogenesis. It starts way before birth, and these oogonia, by the 5th month of fetal life, they have all formed primary oocyte. What does that mean? It means that at birth, we have no oogonia. So, we are seeing that at 5th month we are having these primary oocytes. So what happens to them? These primary oocytes remain dormant. They remain dormant in a girl until her puberty. And they remain dormant in prophase 1. Meaning, primary oocytes enter meiosis, meiosis 1, but they remain dormant in prophase 1. They don't finish the cell division until puberty. So after reaching puberty, these primary oocytes, actually they're always like 5 to 15, they begin maturation with each ovarian cycle, and usually one of these reaches the full cycle, forming a secondary oocyte, and the first polar body. The reason we form polar bodies is that this division is always unequal. So there is always an equal division of the primary oocyte. And the first polar body degenerates, this degenerates. So, and then immediately we shall have this secondary oocyte begin meiosis 2. Promptly it begins meiosis 2, but it is arrested in metaphase. Arrested in metaphase 2, or metaphase of meiosis 2. And it is always arrested there. It is always about 3 hours to ovulation. And it cannot continue the cycle until there is fertilization. We also actually need to air out that we had the primary oocyte having 46 2N, but this secondary oocyte has 23 and 2 and 2N, just like we saw at the end of meiosis 1. So after fertilization, we shall have the second oocyte completing meiosis 2 and forming a mature oocyte. And this mature oocyte, it's going to form mature oocyte and a polar body which is going to generate, and this mature oocyte is going to be 23 single chromosomes and half the amount of DNA. So we can do a recap. We had from maple-brush cells, then we had primordial germ cells giving us Oogenesis giving us primary oocytes giving us secondary oocyte and first polar body. And then we had secondary oocyte giving us mature oocyte and also second polar body. We said that by the 5th month, we have formed all primary oocytes. There is no Oogenesis at birth. So once these start meiosis 1, they are arrested in prophase 1 until puberty. At puberty, it finishes the cycle forming secondary oocyte, and the secondary oocyte is 23 and normal amount of DNA. The oocyte undergoes meiosis 2, and as we said, we have to have 23 and 1N. Those are some of things that are asked in objective questions, and if you've not really understood, you find you cannot answer the objective question. So let's look at the approximate number of oocytes. And let's begin with the primary oocytes. We see that at month 5, we tend to have about 7 million oocytes. Then at birth, we have just like 2 million. We have 2 million, and this means 5M have degenerated. So how about at puberty? At puberty, we only have 40K. Can you imagine? This means that 1.96 M have degenerated. This regards the primary oocytes. How about the secondary oocytes? Secondary oocytes, we see that 12 secondary oocytes are ovulated per year. Are ovulated per year because those are 12 months. One oocyte per month. But of course, that is achieved when a woman is not using birth controls pills. Maybe when she's not on family planning, and when she's not having those anovulatory cycles, when she's ovulating per month. So meaning in the reproductive years of a woman, and we tend to count 40, there are always 40 times 12. So that is a total of 480 secondary oocytes from 7 million primary oocytes at month 5. And let's look at the final process which is spermatogenesis. And we say that spermatogenesis here is formation and development of male gametes. And it is divided into 3 main parts. The first one, we have spermatocytogenesis. We have meiosis, and we have what we call spermiogenesis. Spermatocytogenesis, as you hear, we are forming spermatocytes. Then we are having meiosis, and then we are having, these are morphological changes after formation of the spermatid. We also need to know that this process takes about 64 days, and it takes place in the testes, and it starts after puberty. It's not like oogenesis, which starts before birth. Let's talk about spermatocytogenesis. And in all these processes, we start from those stem cells, primordial, germ cells are going to go to your testes from yolk sac. Remember that side we had these ones coming from epiblast, going to the yolk sac, passing through now to the ovary. So these ones are going to go to the testes. So in the testes, they are going to differentiate into what we call type A, spermatocytes. Remember in your A-level you didn't have these things of type A and type B, but here we have type A and type B. So this type A, spermatocytes, they are the ones that are going to continue supplying stem cells for formation of sperms. They are the ones that are going to keep forming type B spermatocytes, the ones that are committed at formation of sperms. So here they are going to keep on supplying stem cells. They will remain there, and some are going to differentiate into type B spermatocytes. So there are these ones that are committed to forming sperm cells. These ones do not. For them to form, they have to first form type B. And these type B spermatocytes mature into your primary spermatocytes. And all of these are 46 and 2N. Then these ones undergo your first meiosis, and they form secondary spermatocytes. And we saw that these ones are 23, but 2N. And then these ones undergo meiosis 2, and we shall have spermatid, which is 23 and 1N. But these spermatids, they are not yet capable of fertilization. They are not yet good. So they have to undergo post-meiotic changes. And that's where we bring in the third step. Actually here, when we began meiosis, this was meiosis. So here we bring in the third step of spermiogenesis. And spermiogenesis, it is that post-meiotic or morphological changes in a spermatid. And these include formation of a chromosome, they include formation of head, neck, and tail, and we shall also have condensation of the nucleus. That is what mainly occurs in spermiogenesis: formation of a chromosome, condensation of the nucleus, and formation of head, neck, and tail. Most of this spermiogenesis, shall you remember, these spermatids cannot fertilize the ova, so they will need to go through a process called capacitation, and that capacitation occurs in the female reproductive tract for them to be capable of fertilizing the ova. Let's lastly look at anomalies, and here they are main two. We have offspring of older women and offspring of older men. We tend to find that the offsprings of older women tend to have chromosomal abnormalities, and these abnormalities include Down syndrome, trisomy 21. And why? Because there is always increased the incidence of non-disjunction, failure of separation of those homologous chromosomes. And this non-disjunction is attributed to the fact of those dormant primary oocytes being dormant for so long. And some of the clinical findings in these children, they tend to have microcephaly, small heads, they tend to have microphthalmia, they tend to have congenital heart defects, and tend to have protruding tongue, and many others. Whereas these offsprings of older men tend to be born with genetic defect, achondroplasia, and here it's where you find you're having retarded bone growth, and these children, especially the boys, tend to have Marfan syndrome in their paternal age, later their paternal age. Remember, this syndrome is a syndrome of connective tissues. It disrupts connective tissues. Those are some of the anomalies. So that was the spermatogenesis, and I hope that this work can really do you too much good as far as the histology and embryology exam is concerned. And please, if you find the video helpful, subscribe, like the video, and share to your friends, and let's continue learning together. Thank you.